we are really grateful to be part of this incredibly dynamic ecosystem here at McCormick. And I think we're excited to find more. We've enjoyed this opportunity working together and excited to find new opportunities to work together as well. So as we said, I work in service design and design innovation, and Mitra is in neuroscience, mechanical and biomedical engineering. And we decided that the intersection of our work centers around exploring and understanding, quantifying, and designing complex ecosystems. And so those are ecologies that happen both in the natural and physical world, and then in the digital world and that we navigate each day. All right, so with that,
Good.
And that was not an easy intersection to find.
Well, thank you, Amy. So as Amy said, our task when we started working together was to find how understanding what features our brain pays attention to when we touch objects, that's my body of research, how it intersects with how we approach designing interventions in the integrated product service ecosystems that we navigate every day. So this was a challenge. What we came up with was this idea that there's a loop that describes the design process that we both use. So we call it the environmental individual collective action, or ICA loop. And I'm going to give an example using Amy's field of service design. So imagine that a service is started on a college campus. For example, we might accommodate students with food allergies. And so the place or an ecology or the environment includes the campus, dorms, dining halls, sports and clubs, events and parties. And then we have the individual's experience of that environment, so how individual students are making their way through that environment. And then we have the collective experience, which includes multiple stakeholders, including not only students, but their roommates, cafeteria workers, the health center, staff, faculty, custodians, et cetera. And then we have actions that can be taken either by the individual or by the collective, by these different community members, which have an influence then on the environment. And so notable features of this loop are that each element has its own complexity and is hard to model. So of course, each individual is themselves complex. And each element in the entire loop changes or evolves at many different timescales. And external pressures on each element are happening simultaneously. So for example, you can imagine that a particular food substitution in any particular week causes a change in environment, but is also directly affecting a food allergic student. Maybe they have an anaphylactic reaction. And the most really interesting thing to notice about this loop, though, is that this collective experience, the engagement of multiple stakeholders throughout the loop, determines the success or failure of the service. So you can't have a service that just works for one person, right? You need it to work for many of the stakeholders. So I'm now going to look at how the ICA loop relates to my own work in the field of neuroscience. So here we have an environment, and in my case, it might be a field of grass. And as you'll see, I study rodents, I study rats, and so the kind of sensory system that they use to explore that environment, the individual sensors are whiskers, which acquire information about the grass. And then the neural circuits are integrating information from many of these different whiskers, and the neural systems then send motor output to the muscles that control the rat's head and whiskers and body as it navigates throughout the grass, which then has an influence on the environment. Now, if you're a neuroscientist, you're interested in that region of the figure that's over there on the right in the red circle, namely the neural circuits. But there are some problems with just trying to study the neuroscience alone. One of the big problems is that there's over 12 billion neurons in the rat's brain. That's a lot of neurons. And the second problem is that they're connected in complex ways with many different types of electrical and chemical connections between them. But that's not the worst of it. Recent work from a lot of different laboratories has shown that neural circuits that look similar can have very different functions, and neural circuits that look different can have very similar functions. But even that's not the worst of it. Part of the worst of it is that all of these different elements that are leading into the nervous system are really difficult, sometimes impossible to measure. We can't measure the environment. The input from the environment is hard to measure. Sensor dynamics are hard to measure. Muscle and body dynamics are hard to measure. And interactions between the animal and the environment are hard to measure. So the question is, if you're a neuroscientist and you're just sticking an electrode in the brain, how do you know that any of the input stimuli that you're using have any relevance to what that brain evolved to process? It's a huge problem in the field of neuroscience. So this is why you can't just take the brain of a cat and put it into the brain of a rabbit and imagine that they're going to operate the same, right? The pressures on the rabbit have evolved to give it big ears and to have side-facing eyes because it's a prey animal, and its brain has evolved as a prey animal given those sensory and motor systems. And a cat's eyes have evolved to be forward-facing and binocular. It's a predator. It works in the dark. It works typically at night or at dusk. And so its brain has evolved to take advantage of those particular features of the environment. So again, notable features of the ICA loop. Notice that I have here the same the service design loop up in the top right, just to show the analogies with the neuroscience. Each element has its own complexity and it's hard to model. So we've got sensors, muscles, and environmental interactions. We've got each element in the entire loop is changing at many different timescales. In neuroscience, that would be both phylogenetic and ontogenetic timescales. We've got external pressures on each element, which are happening simultaneously. So for example, when the motor system changes, it's going to influence the kind of sensory data that the animal can acquire. And again, the collective experience, that full nervous system, is what determines an animal's ability to perceive and act, and thus ultimately the success of the species. So I'm going to describe two uses of the ICA loop to study the brain. The first is going to be an approach towards modeling this full loop. It's a new approach towards trying to get at the interactions between each element of the loop. And the second is trying to skip over part of the loop here. We're going to try and skip over this individual sensors and directly link environmental statistics to the kinds of circuits that are operating in the brain. So as I mentioned, my laboratory uses the rat nervous system as a model for studying the brain. And in order to understand the work that follows, you should know that all rats, regardless of whether they're here or anywhere in the world, they have about 30 whiskers on each side of their face. They're so common between all the rats that we can actually give them names. They're not very interesting names. The rows are given letters, the columns are given numbers, and the whiskers are shorter as you get to the front of the animal and longer as you get towards the back. And if you've never watched a rat explore, here's an example of whisking behavior. So this is real-time whisking behavior as we set up a rat in our laboratory to explore over different objects. And then in a little bit, we're going to slow it down. And what you can see is that the whiskers are moving back and forth. This behavior is called whisking behavior. You can see the animal is moving its whiskers over this Tinker Toy wheel, and it's going to put its paw right up where its whiskers were. It's determined, how do I need to place my paw given the curvature and orientation of this particular wheel. So it's a very complicated behavior. So the problems, though, with trying to study whisking behavior and how the brain represents whisking behavior is that we can't measure all the mechanical signals at the whisker base. They're too fast, too small, and adding a sensor changes the mechanics. We can't measure the environment. It's complicated. Imagine trying to quantify all of those Tinker Toys. And we can't really quantify the interactions between whiskers and the environment. So those collisions are not possibly to model dynamically, or measure dynamically. So the solution was achieved by a phenomenal graduate student in my lab, Nadina Zweifel. We also worked with Ian Abraham and Professor Todd Murphy, and this work was published in PNAS a couple of years ago. So she built a simulation system that can model this whisking behavior of rats. And let me pause this video. This video automatically started playing, but there we go. So what I'm going to show you here is a video, and it's a complicated video, so let me walk you through the steps. So one of the most common stimuli that a neuroscientist uses to test how neurons in the rat's brain responds is just a bar or a post or a peg moving through the whiskers. And so what we simulated here is this peg moving through the whiskers. So that's what you're going to see in this frame. And then over here, you're going to see all of the whiskers arranged in their regular array, and we're going to be plotting the forces that are exerted at the base of each of these whiskers and the moments. That's the M up there in red. And a moment is just a torque. So if you imagine a teeter-totter with the rotations of a teeter-totter and the torques of the teeter-totter, that's what's being plotted in red there. So we're going to show the forces and the torques, or the moments, at the base of each whiskers, and the magnitude of them is going to be indicated by the magnitude of the vectors that emerge from the base of these whiskers. And then down here, what you're going to see is time is on the x-axis, and on the y-axis, you're going to arrange the whiskers by their rows. Remember I said we could give the whiskers names, so we're going to have the A row, B row, C row, D row, and the columns within each of those rows. And we're going to plot out the magnitude of the moment, the magnitude of that torque, as a function of time at the base of each of those whiskers. We're just going to run the peg through once, and we'll do it then multiple times. The video will loop to do it multiple times in a row. So I really like this video because it's making the invisible visible. We could never measure this directly in the real rat, and we couldn't even possibly monitor each of these whiskers at this resolution. These are really tiny. The whiskers are only about 100 microns at the base, about the size of your hair. Right? Okay, so that's a typical experiment that a neuroscientist might do, but remember that this whole thing is about ecologies and the environment, and so the environment really matters. And so what Nadina did is she went out to Chicago, which was recently voted the rattiest city in the United States, and she took 3D scans of alleyways and dumpsters and such. And so what you see here over on the right is an alleyway with a drain pipe in it. And this video is going to zoom in on the drain pipe, and you're going to see this rat whisking over the drain pipe in its natural environment. And the important point here is that these are the kinds of signals that the rat is actually obtaining in the natural environment. These are the signals that the brain evolved to process, right? That's the key point here. So, and it's the same, what you're seeing the same things in each of the three plots. And I'm not sure how visible it is, but you can see that as the rat's exhibiting this rhythmic whisking behavior, you get these peaks in the bending moments, or the torques at the base of each of these whiskers, coinciding with the times that the whiskers are making contact with the drain pipe. Some of the little whiskers, the reason there are discrete patches there is that the little whiskers are missing the drain pipe because they're on the inside, right? And so this is an example of how it's only the large whiskers that are sending input to the brain, and the little whiskers, aren't making contact hardly at all. All right, so the brain is in continuous interaction with the environment, and what we can do now with this simulation tool, I can imagine running millions or hundreds or millions of billions of simulations to characterize the input that the brain evolved to process and control. And the field is recently calling this a set of synthetic data that we can then use to train a whole bunch of families. We can look at how this synthetic data inputted into families of neural circuit solutions that could achieve the observed behavior, right? So the brain is not going to be identical from rat to rat to rat to rat. That's just the same way that none of our brains are exactly identical. So we need different families of solutions to try and look at how the same behavior might be achieved. So essentially what we're doing is we're building a rat brain from the outside in. Okay. Very briefly, and this is just a preview because this work is not published yet. This work is work that's in review at Nature Communications. And again, the lead author is Nadina, so really a phenomenal thesis done here. But this work was also done very closely in conjunction with Professor Sarah Soya, who was the computational lead on the project. So the question here is as follows. We talked about this loop, but there is a reality out there. Regardless of whether you're a rat or a human or a frog exploring that grass, the grass has properties that do not depend on the animal that's exploring it, right? The grass is the grass, right? It is just the grass. So the question is, what are the properties of the grass that might be the statistics or the properties of the grass that the brain might encode? And so the question is, can we go straight from the environment straight to making predictions about what the brain should encode? And I really only have time to give you a preview of this, but what are the features of the environment that all of these brains evolved to process? So in order to answer this question, what Nadina did is to scan 96 different natural objects, ranging from a walnut, a feather, a twig, a pine cone, a mushroom. And then she made surface patches over each of these different objects. And so these are some example patches. Right now they're discrete, but they were more continuous in the real set of data. The patches were about fingertip sized, which is also the size that a whisker array would cover. We had 96 objects and 128 different patches per object for over 12,000 patches. And then what we're going to do is we're going to take each of those patches and we're going to put it down at a location on the object, and we're going to quantify the distance at every point under that patch to that object, right? And then also on that patch, at each point in that patch, that object has a local slope. There is a slope at that point, and there is a curvature at that point. Right? So we take a patch, we quantify the distance, slope, and curvature at each of those locations, each of the points on that patch locations. And to give you the take-home message of all of this work, curvature stood out as a particularly important metric. And by analyzing curvature, but not distance or slope, we were able to replicate some features of how touch-sensitive neurons in the real primate brain responded to tactile data. So this work is in review, but it does seem that we're able to go straight from environmental features to making some predictions for how the primate brain should respond. So this picture shows that we're going straight from the environment to the circuits, skipping the individual sensors. And now Amy is going to tell you a little bit about how the ICA loop is being used to design services that help people actually do things.
Thank you, Mitra. It has been so much fun getting to meet you, getting to know you a bit, learning about your work. And I'm sure that you're all on the edge of your seats trying to figure out how we might be able to tie these things together. Right? Let's see. We start, I think we can go right to my next slide. So we've said that the ICA loop is really what we've decided kind of ties our work together. So I'm going to talk to you a little bit about how that works in the context of service design. So some of you may be familiar with the idea of service design. You may, just a quick show of hands, does service design as a discipline sound familiar to folks? Okay, well, the idea is, frankly, that services, just like any kind of physical product that you would engage with, need to be designed or can be designed. And it can be hard thinking sometimes about what a service is, because really the modes of services can take lots of different forms, right? We can have interpersonal services, we can have digital services, some services are events, some services are communication plans. They can take all sorts of different formats, but really when it comes down to it, an easy way to think about services is that services help people do things in this context. And so service design is a discipline within design innovation committed to helping design of ways for people to do things. And I've had the pleasure of being involved in lots of different collaborative and participatory design-based projects where we were designing ways for people to do things like navigate transportation futures or access rescued charitable food, learn about different healthcare professions, or even build community while waiting in line. Right. But to give you an idea of what this looks like in practice and how it relates to the ICA loop, I'm going to talk a little bit about a series of projects that I've been working on over the past 7 years with CFAR, the Center for Food Allergy and Asthma Research here at Northwestern, that's led by Dr. Ruchi Gupta. And I'm just going to acknowledge that some of the things I'm going to be discussing are potentially life-threatening medical conditions. And I will be referring to them as disability because that's how the ADA recognizes them. So if that's uncomfortable for anybody and you prefer to take some time, we understand that for sure. But prevalence of life-threatening food allergy among college students in the US is estimated at about 8%. And there's no treatment for food allergies. So what that means is it's really incumbent on the student in most cases to both avoid the food that they're allergic to and make sure that they have their rescue medication with them at all times. And really for some students, being able to do those two things can mean the difference between life and death. So for CFAR and EDI, our collaborations happened over a number of different projects, 2015, 16, and 17, where we were looking at different ways that we could understand and quantify and design into this whole ecology. So over the course of 3 years, nearly 80 students tested and designed and tested different prototypes and interventions spanning a tremendous breadth of activities that somebody who is going to a new college or university might encounter, okay? And in 2018, we published the first academic paper looking specifically at the research and design of these interventions. So this is kind of exciting to share. But today, many of those same interventions that have been prototyped by the students and in these different projects are happening on campuses across the country. So from signage and cafeterias and activities to different things happening during welcome weekends, information that folks are getting in advance, different clubs on campus. The impact has been really broad. It's been adopted in many ways by different food service companies on college campuses. And this is a really big ecosystem. So while there's still lots of challenges in this space, we've seen these interventions positively impact the lives of hundreds of thousands of students across the country. The ecosystem, though, is really quite complex. So at the start of our talk, Mitra introduced the ICA loop, and she gave this example of how this kind of seemingly inconsequential action, right, maybe changing what ingredient would go in the granola today, could really could cause a student to have an anaphylactic reaction or potentially a fatal risk. And so thinking about that in this ecosystem can be helpful. And Mitra, thank you so much for laying out all of the different things happening in the environment and the different places that this was happening. So complex as an ecosystem to start, but then also important to recognize that this is happening across over 1 million food allergic children going to college in the US. Hundreds of people are engaging with those students at each campus. And there are more than 5,000 campuses, each with their own dining halls, dorms, activities, and so forth. So as designers, the challenge that we faced was how might we design different interventions or actions that could positively impact or create a set of constraints that would positively impact these actions and make for a loop in which these students could be successful. So where did we start? We started with Joe. Joe is a student here with life-threatening food allergy. In human-centered design, we often really zoom in to look at a specific person or persona and really try to understand what that person's experience is. And what we learned is that in high school, many students with this disability have a strong infrastructure in place. They may be parents, friends, family, pediatricians, EMT, but when they're transitioning to college, they need to come up with a way to rebuild that infrastructure. And back in 2015, there were not a lot of interventions to help support students through that process. So so the process was very individual, and it was really incumbent on the student to navigate that. But we've all been listening to Mitra. We know that the ICA loop illustrates that these ecosystems are very, very, very complex. And we know from Mitra's talk that everything happening in that system is not just about the rat, right? So to think that this one student would be able to independently navigate this whole ecosystem without other affordances along the way was really not something that was going to be able to be possible. So whether you were a biomedical engineering neuroscientist or not, you understand that this student was part of a much broader ecosystem. So in order to understand that ecosystem and in this new environment and scaffold ways for that student to both navigate safely and with appropriate emotional supports and a sense of belonging in their new environment, we had set out a set of interventions, and those interventions came to be known as Spotlight or Campus Spotlight on food allergy. So from the onset, we engaged participants familiar with the process of going to college with a disability, and because we wanted to better understand who are all of the people that the students are engaging with. We met with students, we talked with folks in all sorts of different offices on campus, different community stakeholders, and that in human-centered design is the process of qualitative research. We mapped the stakeholders, so in service design, there's a tool called stakeholder mapping, which helps kind of understand who all of the different stakeholders are in the system and how they relate to each other. And we were able to determine that there were more than 10 different types of individuals that were likely to be giving day-to-day support to that student with food allergy. There were more than 5 different services that were committed either to health or to emergency rescue that the student would be engaging with, and then service providers across all of those different services. And then more than 5 different health or campus-related support services and all their service providers that the students would be engaging with. And from that, we had these insights and determined where the best places in that loop and it makes sense to make our interventions. So which overall interventions might be the most impactful? The ones that were determined through the work were the notifications of others in the student network, establishing clearly defined roles and responsibilities, and then overall increasing awareness of food allergy signs and symptoms. Those were the overarching areas of opportunity. And then we looked at very specific components in that collective experience. So what points in time, what people, and what roles should we explore? And we narrowed in on preparing for college, orientation, engaging with activities, sports and clubs, external vendors who might be coming to campus, and then also emergency response. So we use service design tools throughout this. We recognize that within a group of seemingly like stakeholders, such as students with food allergies, we know that individuals' behaviors are going to vary really dramatically. So we used behavioral archetypes to try to understand that. And then as we mapped the different journeys that students would take through these experiences, we really tried to understand what those different behavioral archetypes would choose to engage with. There may be one student who really didn't want to engage in all of the different steps and precautions and building their network individually on their own. And there may be students who wanted to go about doing all of that, creating those relationships and building those networks. And when we were designing interventions then into the EICA loop, what we were trying to do is accommodate for all of those different individual possibilities, as well as the collective context and all of these different roles and stakeholders, so that the interventions or the things we designed, so particular actions would be taken, would become standard in the ecology, and that loop would continue. So we know that this is complex. We know that the loop is occurring at multiple simultaneous timescales, that there are so many different stakeholder groups, and we know that it's really difficult to map every aspect of the loop. As service designers, we're designing these individual interventions, and we're hoping that these interventions work. There are two really significant challenges that we have in service design, and one of them is recognizing that this loop is just circling and circling and circling and growing exponentially. How do we figure out how best to measure a specific intervention to know if that intervention is actually serving the entire loop effectively? So that's one of our big challenges. And another challenge that we have is we often call them stakeholders in service design, but they're all of the people that we're designing for, right? Student with food allergies, and then everybody on campus who's engaging with that student. How can we make sure that all of those different participants are really being incorporated throughout the process in a way that acknowledges both their individuality and is very equity focused? So stakeholder equity and measuring impact are two opportunity spaces that I have found. Thinking about the ICA loop, working with Mitra, thinking about the ICA loop and thinking about it in the context of service design. So in service design, HMW, or how might we's, are ways that we sometimes try to understand what new opportunity spaces are. And I think there are two really good opportunity spaces that have really come out of this partnership and the conversations that we've had that could both help us understand how this loop could encourage us to better include and advocate for all of the people going through that system, and how an understanding of this loop might help us better measure those individual interventions. So, what are you going to tell people this talk was about? So, it was exciting and fun and difficult sometimes, but always in a very benevolent way, right? To try to come up with these different intersections, how food allergy and these stakeholder groups and this understanding of the ecology could lead to different frameworks that we can use in different disciplines to view the work that we do in some really interesting and different ways. So we want to thank you. We want to thank you all for being here. We want to thank all of the folks at Segal who supported making this happen. We want to thank Dina Tino for really instigating these different interactions and coming up with a forum in which we could begin to consider these different possibilities. And we want to thank, of course, all of the students and collaborators and other folks that have been so instrumental in the work that we each do. So with that, we'd love to hear your questions. There might be questions.
We left enough time for questions. I think we have like 4 minutes till 1:50.
I'll go ahead and bring the loop back up, just in case anyone wanted to see it. And I think I want to acknowledge that sometimes these really seemingly obvious things, like of course these things all interact, sometimes they can be difficult to come to, difficult to visualize, and it really just helps to have other perspectives and thinking about what you do in the work. So, yeah.
Can you talk a little bit about the directionality of the loop? And is there anything that you discovered around in both contexts that and was interesting?
Sure. I mean, so what I can say in terms of the brain evolution is that all of those pressures are simultaneous, right? If you have a mutation that affects a sensory system, the motor system may have to adapt. And you can also have a mutation that affects the motor system, and the sensory system has to adapt. And so by this process of random mutations in sensory and motor systems, you're pushing the brain one way or the next. That's how a cat developed into a cat, rabbit turned into a rabbit. So the directionality of the loop, I mean, we drew it one particular way in the neuroscience field, but it's bidirectional and it doesn't go one particular way. All of the pressures are simultaneous.
Yeah, okay.
Great. Yes.
Anything in service design to say about that?
Oh, it's all always happening all at the same time. So
directionality doesn't really
the
matter.
directionality is not as relevant. So I think if this were to have arrows on both sides in the context of service design, that would be equally appropriate. Yeah, and every way.
I mean, the idea of the arrows is that a change in one will affect the change. It will affect all of the others. And I couldn't think we couldn't think of a better way to draw it other than directional arrows. But
thank you.
Thanks.
So I really enjoyed that and trying to think about the overlap. One of the things that struck me is that in both of your worlds, there are parts of this loop that are exceptionally complicated, where to the point that it hardly makes sense to try to model it. Mm-hmm. You talked about the 12 billion neurons and how different structures may behave similarly and vice versa, and you emphasize throughout the complexity of the ecosystem. And yet you both model these things. You built the rat brain from the outside in, and you showed us all sorts of models, journey maps, and personas, and so I'm curious what insights you've gotten through the years about how and when to use models for things that arguably aren't modelable.
I can speak to that. So we have a simulation system that can now do the outside of the animal, essentially, right? And so the question is, what about the very first neurons? Here, I should take off my mask. What about the very first neurons that respond to that sensory input? So what you can do there, since you can go and record from those very first set of neurons, and you can model the statistics of that set of neurons. And if you got those primary sensory neurons sort of in a modelable space. And although I didn't have time to talk about it, that's another one of our papers. We published it a couple of years ago, again in PNAS. And so we have the statistics of what are the mechanical signals that those primary sensory neurons are responding to. And so we've moved from the outside of the animal now to the encoding at the primary sensory level. The next stage then is to find the fastest way out of the brain back into the external motor loop. And it turns out that reflexes in the rat whisker system are very fast. So we can complete that loop in only 2 or 3 synapses. So although there are 12 billion neurons in the rat's brain, there are much fewer neurons and a much more regular circuitry within that reflex loop that completes sensor to brain to motor action very quickly. And that reflex loop, I think we can model, and a reflex loop has enough empirical constraints on it that we can start making families of models for that particular loop. And then from there, we go to the next, right? So we gradually build on up to the 12 billion neurons by taking...
Is that what you think is going to happen?
Yes.
Okay. I like it. You own it.
Yes, I mean, and the reason I say that is that this has been my... This simulation work that Nadina did and the recording from the primary sensory neurons has sort of been my vision for the past couple of decades. And I had this vision and we did it. So now let's go do the second order loop. I think we can do it.
I love that optimism. And I was just to tie it back into service design just a little bit. I think these families of models are what help us teach students and engage with folks and look at different problems through similar sorts of lenses. So I don't think that the models are going to include, or will, in my case, will be able to measure everything at any point, but I do think that they give us some different frameworks that can be shared across different disciplines.
Yeah, I would just build on that by quoting George Box, who said, all models are wrong, some are useful. So I think you use a model when it's useful, and I think you don't when it's not useful. So I don't use a lot of models. I think it's important to have them as like, hey, these are tools in a designer toolkit. If this doesn't really serve any purpose, don't use it.
Absolutely. Yes, that is for models in the design space, for these different frameworks and tools that we use, they're just ways of thinking about these different things. We're trying to share them out with other folks. So not prescribed.
Yeah, if it's useful, use it. If it's not, don't use it.
Awesome. Anything else? We hope you've enjoyed lunch. We hope you've enjoyed our time with us. We hope that this has you thinking about some other intersection that you are excited to explore with somebody else here in our McCormick ecology. So, thank you.